Biochemical aspects of hormones. Body activity is coordinated by various chemical messenger systems like neurotransmitters released by axon terminals of neurons into the synaptic junctions, act locally to control nerve cell functions, GABA like that, acetylcholine, GABA, etc. Then endocrine hormones released by glands or specialized cells into the circulating blood influence the function of cells at another location in the body. The neuroendocrine hormones secreted by neurons into the circulating blood. So influence the function of cells at another location in the body. Oxytocin, ADH, paracrines and autocrines. Hormones are classified according to the distance over which they act. Endocrine hormones, insulin, epinephrine. Paracrine hormones, local mediators. IL-1 from macrophages that stimulate the bound T cell to proliferate and differentiate. Endocrine cells, paracrine hormone molecules. Endocrine cells, bloodstream, autocrine hormone molecules, target cells, you could see that. Autocrine hormones. Paracrine in the sense neighboring cells. Autocrine into the same cell. Autostimulatory release of IL-2 enhances the response of T cell to IL-1. Endocrine glands and hormones, hypothalamus, adrenal medulla, adrenal pituitary, posterior pituitary, adrenal cortex, pancreas and intestine, ovary, thyroid, placenta, and kidneys. Once again, hypothalamus, pituitary gland, pineal gland, thyroid gland, parathyroid glands, behind the thyroid gland, thymus gland, pancreas, kidney, stomach, adrenal glands, adipose tissue, small intestine, ovaries, and testes in case of male. So hormones of the three chemical classes, proteins and polypeptides. Hormones secreted by anterior and posterior pituitary glands. Hormones are of three chemical classes, proteins and polypeptides. Hormones secreted by anterior and posterior pituitary gland, insulin and glucagon, parathyroid gland. Then steroids, adrenal cortex, cortisol and aldosterone, ovaries, estrogen and progesterone, testes, testosterone, placenta, estrogen and progesterone. Derivatives of the amino acid tyrosine amines, thyroid, thyroxine and triiodothyronine, adrenal medulla, epinephrine and norepinephrine, dopamine, prolactin inhibiting factor. There are no other known polysaccharides or nucleic acid hormones. Growth hormone and insulin are peptide hormones. Synthesis, storage and release of peptide hormones. Prohormones, endoplasmic reticulum. Prohormones, Golgi apparatus, secretory vesicles. Prohormones, hormones. Stored stimulus, exocytosis. Nucleus, DNA, transcription, mRNA synthesis, packaging, translation, endoplasmic reticulum, packaging, Golgi apparatus, secretory vesicles, storage, extracellular fluid, secretion, stimulus. Steroid hormones are derived from cholesterol. They are not stored. Cortisol, aldosterone, testosterone and estradiols. Amine hormones are derived from tyrosine. Amine hormones derived from tyrosine. I just mentioned earlier. Yeah, not in this video, some other video. The transport of hormones, water-soluble hormones, peptides and catecholamines, dissolved in the plasma and transported to target tissues where they diffuse out of the capillaries into the interstitial fluid and to target cells. Steroid and thyroid hormones mainly bound to plasma proteins. So water-soluble hormones, peptides and catecholamines dissolved in the plasma and transported to target cells where they diffuse out of the capillaries into the interstitial fluid and to target cells. Then steroid and thyroid hormones are mainly bound to plasma proteins. Usually less than 10% of steroid or thyroid hormones in the plasma exist free in solution. I am going to read this once again. Water-soluble hormones, peptides and catecholamines dissolved in the plasma and transported to target tissues where they diffuse out of the capillaries into the interstitial fluid and to target cells. Then steroid and thyroid hormones mainly bound to plasma proteins. Usually less than 10% of steroid or thyroid hormones in the plasma exist free in solution. Protein-bound hormones cannot easily diffuse across the capillaries, hence biologically inactive until they dissociate from plasma proteins. Bound forms act as a reservoir. The two major types of control of endocrine gland function. Hypothalamic pituitary target gland systems, freestanding endocrine glands, parathyroid islet cells release hormones that stimulate the target tissue to produce an effect which in turn modifies the function of the gland. A rise in serum calcium or a fall in blood sugar. Hypothalamic pituitary target gland systems. CNS input, hypothalamus, releasing hormone, pituitary, tropic hormone, peripheral gland, hormone, target. Effect. Then varied out input, freestanding endocrine gland, hormone, target, effect. Okay. Hypothalamic pituitary target gland systems, freestanding endocrine glands, parathyroid islet cells release hormones that stimulate a target tissue to produce an effect which in turn modifies the function of the gland, a rise in serum calcium or fall in blood sugar. In hypothalamic pituitary target gland systems, higher brain centers, feedback inhibition, CRF, TRF, GNRF, GRF, somatostatin. Central nervous system is also there. Feedback inhibition will be taking place. Feedback control of hormone secretion. Negative feedback, positive feedback, not very common. Estrogen from ovary, luteinizing hormone from anterior pituitary, estrogen from ovary. Cyclical variations in hormone release, growth hormone surges during early stages of sleep. Cortisol level peaks during early morning. Functions of the endocrine system: maintain homeostasis, insulin and glucagon maintain the blood glucose level within rigid limits during feast of famine. Then vitamin D, PTH and calcitonin regulate calcium homeostasis, respond to a wide variety of external stimuli. The preparation of fight or flight engineered by epinephrine and norepinephrine follow various cycles and developmental programs. Functions of endocrine system: maintain homeostasis, respond to wide variety of external stimuli, show various cyclic and developmental programs, maintenance of diurnal rhythm, growth and differentiation, digestion and absorption of nutrients. Next major topic is about endocrine signaling. Okay, hormone signaling. Three types of signaling: autocrine signaling, paracrine signaling, types of signaling: autocrine signaling, target sites on same cell, extracellular signal receptor. Paracrine signaling, secretory cell adjacent target cell, endocrine signaling. So blood vessel, hormone secretion into blood by endocrine gland, distant target cells. Hormones are of two classes based on the location of their receptors. We mentioned earlier this classification. Class 1, lipophilic hormones, intracellular receptors. Then class 2, peptide and catecholamines, membrane receptors, generate second messengers. Okay, so class 1, steroid hormones, cytoplasmic receptor, nuclear receptor, DNA, mRNA, protein synthesis. But in case of 2, catecholamines, peptide hormones, membrane receptor, second messenger, phosphorylation enzyme, regulation. Signaling in class 2 membranes involve 5 steps. Which are those 5 steps? Release of the primary messenger in the sense hormone. Reception of the primary messenger, delivery of the message inside the cell by the second messenger, that is transduction. Then activation of effectors that directly affect physiological responses, termination of the signal. Receptors for class 2 hormones located in the plasma membrane. They can be G protein coupled receptors or GPCRs, ion channel linked receptors and enzyme linked receptors. G protein coupled receptors, ion channel linked receptors, enzyme linked receptors. G protein coupled receptors or GPCRs. All these come under receptors for class 2 hormones. GPCRs, ion channel linked receptors, enzyme linked receptors. First picture depicts G protein coupled receptors. Signal binds, receptor, G protein binds, G protein activated, then some sort of biological action will be taking place. And channel linked receptors, ion channel linked receptors. Signal binds, channel opens, ions flow across membrane. Then signal enzyme linked receptors. Signal binds, enzyme activated, enzyme inactive, enzyme activated, enzyme generates product. Then G protein coupled receptors has 7 hydrophobic plasma membrane spanning domains. Serpentine receptors, hormone binding site is extracellular coupled with the heterotrimeric GTP binding protein in the cytoplasm. So this has learning about gene protein coupled receptors has 7 hydrophobic plasma membrane spanning domains. Serpentine receptors, hormone binding site is extracellular coupled with heterotrimeric GTP binding proteins in the cytoplasm. G proteins are their receptor alpha subunit and beta gamma subunit. GDP binds to alpha subunit. Binding of hormone to the receptor activates the G protein. Then binding of hormone to the receptor activates the G protein. Receptor G protein inactivate GDP hormone G protein active GTP. GTP activated target protein, which is an enzyme. Then extracellular fluid and cytoplasmic regions are there. There are different classes of G proteins. GS stimulates adenylyl cyclase converting ATP to CAMP. GI inhibits adenylyl cyclase in decreasing CAMP levels. GQ activates phospholipase C, PIP2, DAG and IP3. The target protein and subsequent actions vary depending on the class of G protein activated. Neurotransmitters, receptor, G protein, effector protein, second messenger, later effectors, target effector. In case of norepinephrine, adenylyl cyclase activated, protein kinase A, CAMP, increase protein phosphorylation. Then in case of glutamate, phospholipase C, diacylglycerol, IP3, protein kinase C, Ca2 plus release. Increase protein phosphorylation and activate calcium binding proteins. Then dopamine, same case happens and increase protein phosphorylation. Classes and functions of G protein, not that much necessary. Then second messengers. Molecules that relay signals from receptors on the cell surface to target molecules inside the cell, in the cytoplasm or nucleus. They can be hydrophobic molecules, DAG, IP3, hydrophilic molecules, CAMP, CGMP, IP3 and Ca2+, gases NO, CO and H2S. Consequence, the hormone signal may get amplified significantly. They can diffuse throughout the cellular compartments and can influence various processes. Multiple signaling pathways can use some second messenger leading into crosstalk. Then actions of CAMP, hormone receptor, adenylyl cyclase. Several hormones act through calcium or phosphatidylinositols, oxytocin and vasopressin. NO and ANP has CGMP as the second messenger. Insulin and NGF have receptors with tyrosine kinase activity, enzyme-linked receptors. Insulin-bound receptors sites, tyrosine kinase domains. Ion channel-linked receptors. And the number and sensitivity of hormone receptors as regulated. The number of receptors in the target cell usually does not remain constant. Down-regulation of the receptors, increased hormone concentration and increased binding. Increased hormone concentration and increased binding with its target cell receptors sometimes cause the number of active receptors to decrease. By inactivation of some of the receptor molecules, inactivation of some of the intracellular protein signaling molecules, temporary sequestration of the receptor to the inside of the cell, destruction of the receptor by lysosomes after they are internalized, decreased production of the receptors. In each case, receptor down-regulation decrease the target tissue's responsiveness to the hormone. Then upregulation by receptors. The regulating hormone induces greater than normal formation of receptors or intracellular signaling molecules by the protein manufacturing machinery of the target cell or greater availability of the receptor for interaction with the hormone. When this occurs, the target tissue becomes progressively more sensitive to the stimulatory effects of the hormone. Then summary, class 1. Cytoplasmic receptor, glucocorticoids, estrogen, progesterone, testosterone, nuclear receptor, vitamin A and thyroid hormone. Class 2, ion channel linked receptor, enzyme linked receptor, insulin receptor tyrosine kinase, GPCR. GS stimulates adenylyl cyclase, GI inhibits adenylyl cyclase, GQ activates phospholipase C.